Today in the class, we will learn about the DNA and relevance of knowing DNA. Other learning objective of this class would be what do genes do and what is the central dogma of molecular and biology. Okay, because you have been learning about the cell, the nucleus. Nucleus you have learned has chromosomes, and chromosome is basically DNA and protein. And what is the structure of DNA? How did we identify that DNA is a inheritance biomolecule? All those things you have learned in the previous classes. And in this class, we will also study a little bit more in detail. What are the genes? What are the DNA and its relevance? Why you should know about the DNA. Take an example about imagining the future. So let us imagine that it is the year 2050 and you have set IIT as your goal for your undergrad degree. Okay? You enroll in one of those coaching classes that have an excellent success rate for cracking the JEE. Correct? The first day you show up in your coaching classes and you are asked to give a sample of your blood, which is given to a genetic testing laboratory to test your intelligence level or the genes that participate in IQs. And those genes are mainly telling basically your intelligence level and ability to be an engineer. Okay. After 2 days, the results of your genetic test shows that you have a 90% probability of being an outstanding engineer. And then you are admitted to the IIT Bombay immediately. So can such tests be available? And how you do those tests? Can the genes be responsible for the IQ? Some of you might have heard that yes, genes for intelligence are known now. Some of you may be knowing that there are many things now you can identify by knowing more in detail about the genes. Another good example about why you should know about the DNA or why knowing the DNA can solve the mystery. And we will discuss very briefly a story about criminal investigation. And some of you might have also heard about forensic science, where they really do the investigations, even the criminal investigation, by knowing the DNA. And to add up to this story about how DNA can solve the criminal investigation, we will discuss little bit in detail about this story of Earl Washington, who was in 1984 convicted for the life for rape and murder. But after 16 years, this gentleman was found to be innocent based on the DNA evidence. And how you do that, what in the DNA you look, those details we will discuss. And these details, if you are interested, you can also find in your Campbell book in figure 20.25, but we will discuss in detail in the class as well. Now think about the eukaryotic genome as instruction manual, because this is the genome. Basically, it's a instruction manual and tells us about all the instructions, whatever we do. I have been mentioning you in the last classes that parts like viruses, they have very small genome. E. coli, like bacteria, they have larger genome, at least larger than the viruses. As you go to the single cell to multi-cell species, the genome sizes generally become larger and larger. Now, if you think about humans, this has 3,000 megabase. And if you want to write this instruction manual in this encoded language of APGC, then you can fill lot of 80 volumes. It's that large. If you just simply write all the letters, these many manuals will be filled. Basically, what we want to say here, that human genome is complex and it has very large number of base pairs. think about our DNA, which is basically a biomolecule, and it stores the information in the form of the nucleotide base pair like ATGC. And this particular instruction manual is a long-term stable hard copy, because you must have heard DNA is very stable molecule and it can be even isolated from the fossils. That means it is very, very stable. And for the long-term stability, this can be considered as the hard copy of the instruction manual. Okay, then this is a hard copy and it is stable. That's why we do not see a lot of changes in the DNA structure as we grow. But to make the building blocks of our cellular functions, we need proteins. But proteins are called, which you see on the right, this black color thing. And this is protein, and proteins are building blocks and machinery of the cells. To make the proteins, which will ultimately do the function, this instruction manual makes something called RNA by the process called transcription. We will learn it, how the DNA makes RNA. The process is called transcription. This RNA is very unstable and very short term. So, and this RNA, you can think about cache memory. Once RNA is formed, RNA makes the protein, which as I said, which are building blocks and they are machinery of the cell. They programs the cells, they do all the functions. So again, DNA is very stable, works as hard copy. Then it, by the process called transcription, DNA makes RNA, and RNA is the unstable short term, act as a cache memory, and the proteins which are formed by the process called translation, and these are the programs of the cell. Overall learning objective from this class is about the genome, where information is stored. Little bit again, I will repeat. I am sure that Professor Sanjeeva already told you about chromosome, gene, nucleic acid, and DNA structure, but I will just briefly mention all of them. Then we discuss what is the relationship between genes and enzymes. For example, what are the genes? And then we will mainly focus on the central dogma. Okay. Just to refresh your memory, again, DNA is the heritable material, which is basically present in the chromosomes. And chromosome is a very long DNA plus protein, right? And what is a gene? In biology, you have to define things, define the terminology very nicely, especially what is the gene? Gene is a discrete unit of inheritance. It is basically a part of the DNA. And what is DNA? DNA is a class of compounds called nucleic acids, and they are mainly the polymer of the nucleotides. So nucleotide is a monomer, multiple nucleotides together they make DNA, and those nucleotides, you know, ATGC in case of DNA and AUGC in case of RNA. Okay. What is the function of the DNA? DNA enables living organisms to reproduce their complex components from one generation to next. So when these living organisms reproduce, so every offspring, you must have learned that one DNA, one strand we get from mother and another is from the father. And DNA codes for the genetic instructions in all living organisms and some species of viruses. As I said last time, viruses are the exceptions. They also have RNA as the genome, right? Information is encoded in the DNA structure that programs all activity. So this is the major learning objective of the class, that information is encoded in the DNA structure, that ATGC, in the form of these programs. These 4 letters, the programs are made. And these programs, all the activity, whatever we do, that activity is governed by the DNA structure. And a small DNA structure is genes, which are the discrete unit of inheritance, and which genes will give rise to what proteins, and those proteins will give rise to the function. Let's take one topic at a time. So first question, how DNA is packaged in the cell. DNA is a very, very long molecule. If you see the total length of DNA present in one adult human, you can calculate by length of one base pair multiplied by number of base pair per cell, and you can take an approximation of the number of cells in a body. So 0.34 nanometer is the length of one base pair. Total base pair is around 6,000 million. So 6 times 10 to the power 9 multiplied by 10 to the power 13 cells in a body. So if you see the total length of the DNA in meters would come from 2 times 10 to the power 13, which is far more than the probably how far the moon is. So this long DNA, 10 to the power 13 meters long, is packaged in a very small cell, which is hardly, let us say, 5 to 10 micron meter size. So how this 10 micron meter or 10 micrometer, 10 micron size cell contains such a long DNA molecule? How it is possible? That is only possible when you have multiple layer of the packaging. So we will learn how such a long DNA is packaged in a cell. So first stage of the DNA packaging is that DNA is double helix. So that means there are each 2 ribbons of the DNA molecules, and each ribbon represents a DNA strand. And what you see here, DNA double helix, 2 nanometer in diameter, and it's a 2 ribbon. They are representing each strand of the DNA. Next level of packaging comes from the presence of these histone proteins, which are positively charged. Okay, and DNA is negatively charged because of the presence of phosphate groups in the nucleotides, and the phosphate backbone of the DNA is negatively charged because of the presence of those phosphorus and oxygen atoms. Okay, and this aggregate of histones, because these are positively charged, they resembles a ball of wool, and DNA is winded or twice around the histone, and this DNA and histone complex, what you see here in the TAM image, just if you see this thread, what you see, and the knots, they are basically the each nucleosome. Okay. So once the histone and DNA, they make the complex, the DNA is wrapped. But then second level of packaging comes that involves the interaction of the histone tail. So now the multiple histone proteins, they will interact by the tail region and then give rise to another packaging level. And finally, these DNA and histone protein, once at first, as you said, they were from the head, multiple round of the coiling. Then you saw the tail regions of the different histone proteins. They interacted, make the nucleosome. And now the next level is the formation of loops by these 30 nanometer fiber, because multiple now the histone proteins, they will interact with the tail region and they make the big fiber, which is 30 nanometer fiber. And this is how the complete multiple folding will occur and give rise to the large chromosome. As I said, these different loops, then they will again further coil and they will fold to 300 nanometer fibers, leading to further compaction and ultimately giving rise to the chromosome. So basically, this packaging you can think like a storage compactor, where you arrange the books in a way that in these shelves on a railing, and each bookshelf will have a steering wheel, pack shelves next to each other, and they're easy to move whenever you want to take out a book. So similarly, you will see whenever there's one part of the DNA, for example, that is gene, we need to make the protein from this gene, how the unfolding nicely will happen, and then this gene will be like open for its expression that will make the protein ultimately. Eukaryotic genomes are organized into chromosomes. So as I said, that there are DNA ultimately packaged into chromosomes. Humans have 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of gender chromosomes, either X or Y. Right? These image below shows a spread of human chromosome, and each painted with a different color. On the right, these are organized into a karyotype. So basically, this karyotype or chromosomal arrangement can tell us more about the diseases, such as cancer, because in cancer, there's some aberration in the chromosome organization or in the chromosome structure. For example, one disease I can tell you, such as Down syndrome, where there's a trisomy in the chromosome number 21. Instead of 2, there are 3. And so as I mentioned, that if there is an aberration in the chromosomes, that causes certain diseases. So for example, this chromosomal aberration, basically the abnormal chromosome number, and that is also called aneuploidy. And this happens because of many reasons. One is non-disjunction. So you have studied the meiosis. In the meiosis, you have when these chromosomes divide, they go in one in daughter cell. But sometimes these there are aberrations, and that causes the problem. For example, if instead of 2, if there's only 1, so monosomic, there are 2n minus 1, so there's 1 less. If there is a 1 extra, instead of 2, that's got 2n plus 1, it's called trisomic, which happens in the case of Down syndrome. So 21 chromosome, instead of 2, there are 3. And that risk of having these kind of diseases, they increases with the maternal age. So if the mother's age is very high, with the age, the risk factor of having the baby with the Down syndrome increases. The mechanism by which this chromosomal aberration happens, that can be multiple types. So alteration in the chromosomal structure can happen because of the deletion of certain part of the chromosome. For example, in the figure at the top A, there's a deletion of the D segment of the chromosome, and this chromosome is now shortened. So this can happen actually in the natural course. And when this kind of deletion happens, this can be disastrous as well, or it may not have any impact on the person. Duplication. Sometimes for example, a segment of the DNA, for example, B and C here in this particular example, this repeats. So those can cause the alteration, aberration in the chromosome structure. Another example is inversion. So for example, there is a segment or the DNA sequences A, B, C, D, E, that is the gene sequence, but there is somehow a certain segment of the DNA is inverted. So an inversion reverses a segment within a chromosome, and now it's opposite. Another mechanism by which this translocation, chromosomal aberration happens is translocation. So for example, here in this particular chromosome, there was a sequence like A, B, C, D, E, F, G, H, and then another one, M and O. But during certain issues, so a gene, a translocation moves a segment from one chromosome to a non-homologous chromosome. In a reciprocal translocation, the most common type, non-homologous chromosome, they exchange the fragment. So just remember that what are the different mechanisms chromosomal aberration happens. Another very important example of chromosomal aberration is alteration. Basically, alteration in chromosomal structure is that Philadelphia chromosome. Philadelphia chromosome means in the normal chromosome number 9, you have like this kind of a structure. Normal chromosome 22, there is another normal chromosome, which are very different in structure. But there's a reciprocal translocation where the one segment of the chromosome 9 goes on chromosome 22, and similarly, a small segment of chromosome 22 goes to chromosome 9. And then this translocated chromosome 9 and translocated chromosome 22, and in fact, translocated chromosome 22 is called Philadelphia chromosome. Okay, and this kind of translocation is associated with chronic myelogenous leukemia in one type of cancer. So cancer generally happens because of this kind of mechanisms of chromosomal aberrations. Okay? The cancerous cells nearly in all CML patients, CML is this leukemia blood cancer patient, they contain an abnormally short chromosome, the so-called Philadelphia chromosome, and an abnormally long chromosome 9. What you see here, the chromosome 9 becomes very long and chromosome 22 is very short. Okay? Again, just in very brief, in the chromosome, we have genes, which are like just a small segment of the chromosomes. So what a gene is certain stretches of DNA that code for the hereditary information. It is the hereditary unit of life. And what is allele? Alleles are the alternative forms of a single gene or gene loci. So this you have studied in greater detail in your genetics classes. I will not repeat it, but it's just for the information that in a one chromosome segment, you see the P is just a small part of the DNA, and this is a small part. A very defined part is called a gene. And what is genotype? Genotype is an organism's full hereditary information. Okay, so how, for example, the eye color, you can define this gene as is BB. Let's say, so the genotype, if it is recessive, then small b and small b. If this is a heterozygous or the homozygous for dominant, it is called capital B, capital B. Just I'm trying to basically revise your memory. So this is called genotype. Genotype is basically the full hereditary information of the organism. Phenotype is actual observed property. So eye color genotype can be recessive or dominant, and that you can symbolize it with a small b or capital B. But actual phenotype is either blue or brown eyes. So that could be called the phenotype. Also, what is the chemical composition of the DNA? These are chain of nucleotides linked by phosphodiester bridges. In fact, these are the polymer of sugars, phosphates, and nitrogen bases in water. Okay, and if you see the DNA, what you generally look, this has two strands, like two ribbon-like structure, what I said, and it is always in the aligned as 5' and 3' end. So 5' end is called 5' because very simple reason is because it's a fifth carbon, you have the phosphate ring, and here on the 3' end you have on the third carbon a free hydroxyl group. So the end which has phosphate as free called 5' end, another end that is hydroxyl at free at the 3' third carbon of the sugar, that's called 3' carbon. There are many scientists who try to elucidate the structure of the DNA. First, Linus Pauling hypothesized that there are 3 chains and which are twisted around each other in rope-like strand. Then came the Wilkins and Rosalind Franklin X-ray crystallography data, and that actually for the first time told that there are 2 rings, and these nucleotides which are making these 2 ribbon-shaped structure, they are 3.4 angstrom apart in the chain, and structure repeat after 34 angstrom interval, right? And then came Erwin and Shargov. They told us how many A's are there, how many T are there, how many G and how many C. And very important, which you all know, of course, that percent A is equal to percent T. The number of A are equals to number of T's, and similarly, percent G is also equals to percent C. I'm not going to get detail on these parts because you already know from your, I think, 6th, 7th standard nowadays, you are learning the DNA. Again, just to revise that the great discovery of the Watson and Crick and Wilkins, which led to them a Nobel Prize in 1962. This was the structure of DNA, which they mentioned that it's a double helical structure. What are the distance between each nucleotide? How much is the distance from one loop? All these things these gentlemen discovered. Now let us come to the main objective of this class. The main objective of this class is how the information is stored in the DNA. So how the DNA stores the information, how does RNA, the other type of nucleic acid, fit into this gene expression, which I mentioned at the beginning. So what is gene expression? Gene expression is the flow of information from DNA to protein. As I said in the beginning, DNA is your hereditary material. Is a hard copy of the information instruction manual. And what is protein? Protein is the building blocks and the functional machinery. So how the information turned into the function. So the information is DNA, stored in DNA. Function is done by the protein, and how these proteins are formed by the DNA. This process, this flow of information is called gene expression. DNA synthesizes mRNA, which interacts with the ribosomes, which you learned in your first class, and that direct the protein synthesis. So if you see here on your left, how the flow of information, how the gene expression is happening. So you have the DNA in the nucleus. First step is synthesis of mRNA in the nucleus, and that process is called transcription. This RNA then, with the help of ribosome in the cytosol, that makes the protein. So the second step is movement of mRNA into cytoplasm via nuclear pore. And then now your RNA is in the cytoplasm. And in the cytoplasm, you have the ribosome, where synthesis of protein using information carried on mRNA. So based on the type of mRNA, the new proteins will be synthesized. So in a eukaryotic cell, DNA in the nucleus programs protein production in the cytoplasm by dictating synthesis of mRNA. Okay. So you know in eukaryotes, you have the nucleus, that's why everything happens, at least the transcription happens in the nucleus. RNA protein synthesis happens in the cytoplasm. However, in prokaryote, nucleus is not there. So RNA synthesis as well as protein synthesis happens in cytoplasm. So the main 2 points you remember, what is called gene expression. Second, gene expression is flow of information from DNA to protein. For the flow of information from DNA to protein, you have another unit, RNA, that helps in the protein synthesis. And in eukaryote, again, RNA is synthesized in the nucleus, protein is synthesized in the cytoplasm. So how people come to know at the first time that these are the genes which makes a certain protein, or what genes do what function? What do genes do? At the first time, how people come to know. For example, Professor Sanjeeva told you about the peas, coat color in animals, white eyes of Drosophila, black urine in certain patients, or any gene of the shape of the seed, color of the leaf. How you know that these are this particular gene makes this particular protein, and that giving you that function. So what do genes do? What kind of experiments one should do to show that yes, genes are responsible for the particular function? So let's learn about one particular example, how genes dictate production of certain enzymes in a metabolic pathways. Okay, So you have heard many metabolic pathway, glycolysis, TCA as your 6th, 7th, 8th, or 11th standards, right? There are different metabolic pathways you might have heard of. How the genes dictate the production of enzyme, for example, right? So genes dictate production of specific enzymes, correct? And cell synthesize or degrade organic molecules via these metabolic pathways. And each chemical reaction in a pathway is catalyzed by certain specific enzyme. So think about this particular arginine biosynthesis. This is arginine, how arginine is biosynthesized in the mammalian liver, right? So you imagine first precursor is there. Precursor with the help of enzyme A make ornithine. Ornithine, let's say one intermediate. Ornithine, another enzyme take part and convert it to citrulline. Citrulline again by enzyme help of third enzyme converted arginine. So how do you know that these genes are helping in the enzyme synthesis and converting these metabolic different intermediates into the final product? One of the very good experiment done by Beadle and Tatum in the year 2004, they did a beautiful experiment and published their work in Nature Review Genetics, one of the very, very good journal. And they have shown how the genes and enzymes are related. So Beadle and Tatum approach was that instead of asking which genes are responsible for the observed phenotype, they actually looked for the genes that control already known reactions. Okay? So they knew what reactions are happening and they're looking for the genes that control those reactions. And for their experiments, they chose a fungus, a bread mold. It's a fungus and scientific name is Neurospora crassa. So they used this fungus as an experimental model to look for the genes that control already known reactions. So Beadle and Tatum, then they used a very good strategy. They created one food supplement for the Neurospora where it can barely grow. So they took some food and make this good supplemental food where Neurospora can barely grow. And that food they call minimal media. Minimal because it has minimal components on which the Neurospora and this fungus can grow. So they call it minimal media. And then they make a very good media. Very good media means the food requirement with the bare minimum supplement, plus they added all 20 amino acids in that minimal media. And that media they call complete media. So minimal media that consists of inorganic salt, glucose, and biotin, which is a vitamin, in agar, which is a support media. Okay? And then the second they make all the complete media, which they call consists of minimal media, plus all 20 amino acids and few other nutrients. So the normal Neurospora, normal fungus, which is a normally growing fungus, and when something is normal, we call wild type in the biology. If something undergoes genetic alteration, for example, mutation, we call it mutant. Okay, so wild type Neurospora, wild type fungus, that can grow even on the minimal media because it can biosynthesize all the other molecules required for its growth. So all the genes are working in the wild type Neurospora, let's say fungus, and with the bare minimum media it can grow. Okay. So to find out what genes do, what Beadle and Edward Tatum did, they exposed this fungus to the X-ray. And many of you know what X-rays does. X-ray causes genetic alteration. They change the genetic makeup by causing mutations. That's why I say don't get exposed to too much X-ray. Because X-ray can change X-rays, UV light, they can change your DNA, they can cause mutation. So by exposing them with the X-rays, they isolated some mutants. Mutants means where there are genetic alteration. And they isolated only those mutants which were unable to survive on minimal media. So they took out some mutant and they grow in the minimal media. They were not going in the growing in the minimal media, and those ones they isolated carefully. So here they have wild type neurospora. They have exposed it to X-rays. Now they have got a lot of mutant neurospora, mutant fungus. And when they growing on the minimal media, there was no growth. That means something changed in the genes that not allowing them to grow, right? And then they grow in complete media, which has 20 amino acids. They were growing. Okay, so they isolated only such mutants which grow in complete media, but not in the minimal media. Okay. And what they found? They found a very good thing, that the wild type cells growing and dividing in the controlled minimal media, but mutants are not growing. So what this experiment tells them, that mutant is not able to synthesize one or more essential components. Those genetic change happened in the neurospora that is not allowing them to synthesize their essential component anymore, and because of that, the cells are not growing. And now here they have done the trick. The trick what they have done, all the mutants which were not growing anymore in the minimal media, what they will do, they had these many different components which they added in the complete media, right? So the 20 additional components they added. So they took the minimal media, added that first component. Again, there was no growth. But when they added second component, and let us say it's arginine, they added, and suddenly it started growing, correct? So and then similarly, they added third component, and they knew what third component they are adding, either growing or not growing. So this experiment told them that mutant is not able to synthesize, for example, component 2, but when you give it from outside, they can start growing, indicating that this mutant has defect in the genes that is critical for the arginine biosynthesis. That's how they did this beautiful experiment. Similarly, other scientists also did similar or little bit different experiments to prove that to prove the function of the gene, or the relationship between gene and a particular function. And another example is given in your book, is Adrian Serb and Norman Horowitz. Okay, they also used the Beadle and Tatum's method to investigate the biochemical pathway for all arginine biosynthesis. Okay, and this you should read, because in the next quiz, one question will come from their experiment. So now you learned about the DNA, chromosomes, genes, how to identify that genes are responsible for certain function, right? And what are the experiments done at the first place to show that yes, genes are responsible for certain functions, and the gene function relationship. Now we will discuss what is the central dogma of the molecular biology, right? How the informations are flowing. So information, I said, flowing from DNA to RNA to protein, and DNA to RNA, the process is called transcription. RNA to protein, the process called translation. And now the central dogma is slightly revised, because there is a process where DNA to RNA, but RNA can also do the DNA by the process of reverse transcription. This we discussed slightly in the virus chapter also. And RNA can also make the RNA, and DNA can make the DNA, and RNA make ultimately the protein. So that there is slight revision as we are learning the biology. Biology is constantly changing subject. How transcription works, how DNA makes RNA, and how gene translated into proteins. So DNA to RNA to protein, you might have a triplet codon also, triplet code. So for example, if this is a DNA, so DNA has two strands. One is 5' to 3', another is 3' to 5'. And always remember that the template strand that makes the RNA is the 3' to 5'. So 3' to 5', that is a template strand. And this template strand as a template, and the by process of transcription make the mRNA. And the mRNA has, instead of T, you know that it has U. So A turned into U, C is to G, and UGG, these triplets are called codon. And by mRNA will make protein by the process called translation. And the protein, you know, protein is made up of different amino acids. And one amino acid is coming from three nucleotides, and those three nucleotides are called codons. How transcription work? What are the different stages of the transcription? So if you have these double-stranded DNA, 1 5' to 3', then 3' to 5'. First is process is called initiation. So after an initiation, an RNA polymerase, that is the enzyme that makes RNA. So after RNA polymerase binds to the promoter site of the DNA strand unwind, that means opens up, and the polymerase initiates RNA synthesis at the start point of the template strand. And you already learned that template strand was 3' to 5', right? So, and then, so as you see here, 3' to 5' acting as a template strand, and another strand is non-template strand. Then second step is called the elongation. So polymerase moves downstream, unwinding the DNA and elongating the RNA transcript form. So whatever the transcript is made on the template strand, that would be 5' to 3'. Okay. Third step, once this RNA is synthesized completely, RNA polymerase keep moving upwards, and once it is synthesized, then it terminates. Wherever the gene endpoint is, it terminates. Okay. Eventually, the RNA transcript is released, the RNA and polymerase detaches from the DNA. Okay. That is, makes the complete RNA synthesis. So transcription in the eukaryotic cell is always happening in the nucleus. However, in the prokaryotes, where there is no defined nucleus, the transcription happens in the cytoplasm. Okay. So if this is a bacterial cell, where the DNA will make mRNA in the cytoplasm. Okay. Similarly, once the mRNA is formed, translation will also happen in the cytosol. So our ribosomes are there in the cytosol, RNA will attach to the ribosome and protein synthesis will happen. However, in eukaryotic cells, transcription as well as translation, they are two different compartments. Transcription is happening in nucleus and translation will happen in the cytoplasm. One additional very interesting process happens in the eukaryote as the organisms, as the microorganisms or the organisms, they become complex. Their even processes becomes very complex too. For example, in eukaryotic cell, first the transcription happens, it gives you the RNA. So initial mRNA transcript is called primary transcript. But there are so many processing happens before, so this pre-mRNA and then RNA processing happens, and then it becomes messenger RNA. Processing is mainly for the for certain criteria. Maybe at this stage, at your stage, is not needing what exactly processing is required for, but you can remember that there is a first pre-mRNA is formed, and then the certain processing happens to yield the finished mRNA. And the processed mRNA that goes to the cytoplasm, and with the help of a ribosome, it makes the polypeptides. Once the processed mRNA comes, then there are so many ribosomes, several copies of this polypeptide chains are made simultaneously, because here you can see in that image, there are so many ribosomes, they are binding to the mRNA and making multiple copies of the protein, like insulin hormones or other hormones or enzymes. Multiple copies, lot of amount is made. Some of you may be thinking that there are thousands and thousands of genes, and in our body, if genes are regulating all the functions, whatever we do all the time, are these genes working all the time? do all genes work at a time, or only few genes are working at a time but remaining remain silent, or what makes them work, right? These are very interesting questions, right? Now think about from the egg to this baby stage, right? Egg, here you have egg cell, sperm, they diploid cells become the fertilized egg with DNA from both parents, then embryo cell with copies of inherited DNA, and then you have functionally distinct cell and tissue and organs in a baby. And some of the information stored in DNA is used by all the cells, but each cell uses just the requisite information. Every cell has all the information, but eye cell will only use information required for the eye. Nose cells will only use information that is required for the nose, and rest of the information is never used. Only the information which is required. For example, you might have noticed certain genes only expressed at certain age in your life. Infants have certain genes expressed. Teenagers would have certain expression of certain genes. Once your teenage is gone, then certain genes switch off. So switch off and switch on genes based on the time, based on the stage, is very nicely controlled. So why does a cell contain information that it never uses? So you may think like, why a cell in the eye should have all the information? Why not it just has the information of the eye? We don't have a clear answer. One can speculate and come up with possible answers, right? Another question you can think, how does a cell decode only the relevant information contained in chromosomes? As I said, a cell has big, long DNA. How it decodes that only the information it requires to make, let us say, pigment in the eye. By this process is called regulating the gene expression. So that switch on and switch off switch will decide that which genes will be on, which genes will be off. So the switch on and switch off genes, how gene expression is regulated, this will be, now you will be learning this as in your next tutorial. And how certain genes switch on, what makes them switch on, what makes them switch off, how this expression of the gene, which I called in the beginning flow of information, is regulated. It is mainly regulated by operon, and operon is a beautiful concept, and you need half an hour minimum to learn it. And I have ensured that my colleague will tell you in detail of each component of the operon and help you learn, because that's a very beautiful concept of the gene regulation. You must learn, and you should also have any question. I will be happy to discuss. With this, I thank you all, and this is the end of this class. Thank you.